are very keen to participate, and it's
Okay.
a really nice dynamic group. So I think.
Although you cannot see them, we have already at least 40 participants in this conference.
Yeah.
Of those that appear in the
YouTube channel, yeah, sure.
But I have no information. Just you should have now about 100 persons really eager to hear you.
Well, I hope I don't disappoint 100 people.
No, not at all.
I'm sure we're going to enjoy it very much. Thank you very much again.
Yeah. So we're former postdocs. Dr. Arias is here also, and me from Wapping's group. So we had the fortune to listen to you a couple of times when we had these shared seminars.
Yeah.
And so very nice to have you here now.
Yeah, and since then we have been following your work, and it's very interesting and very inspiring for us to continue the research over here. Thank you so much for giving this talk, Doctor.
Again, it's my pleasure, and yeah, I mean, like I said to Violeta, we should meet in person. We can talk more about opportunities to work together, or email works too, but it's
never the same.
Never the same.
Let me tell you that use is traditional in our institute to wait at least 5 to 10 minutes because people will have troubles connecting or arriving or whatever. So we usually start 10 minutes late. That is completely normal.
Yeah, in Israel, they call that academic time. When you say 1 o'clock, it's really 1:15 or 1:10 academic time.
Okay, perfect. Academic time.
Yes. Because academic.
So we can start introducing you if you want. Now there is around 30 people in YouTube and 50 people here, so it's a nice audience already. So welcome everybody. Today it's my pleasure to introduce our speaker. Dr. Buxbaum is a director of the Center for Autism Research. He is the vice president of the psychiatric department in the Mount Sinai Hospital, and he's the founder of the Autism Sequencing Consortium, who is now has sequenced around 60,000 individuals with autism. Dr. Buxbaum has been a pioneer in the integral study of autism, and with more than 300 publications and almost 70,000 citations on his work, he is a leading expert on the topic. So we are very pleased to welcome Dr. Buxbaum, and the stage is yours. Thank you so much.
Thank you. First of all, thank you for the invitation, and thank you for everybody who's joining. I'm afraid it's going to be a very molecular talk, a genetics-focused talk, but hopefully it'll be interesting as well. And I really, again, want to thank Violeta for inviting me and putting this together. And I'm going to try and share my screen, or not my screen, but my slides. And what am I missing here? What am I missing? I'm missing that. Okay, good. Couldn't see it. All right, so I'm going to talk a lot about gene discovery and autism and how it's kind of, maybe changing the thinking about therapies, but certainly also, in an obvious way that I'll talk about only briefly, changing kind of the preclinical research that people are doing in autism. And again, I really do want to start from the beginning to thank Violeta and all the participants. not only from me, but from the Seaver Center. We really welcome the opportunity to meet other groups and exchange ideas and find ways to collaborate, even in the time of Zoom. So thank you for the invitation. And if you have questions, if something's not clear, by all means, you should try and interrupt. I don't know how you'll do that, but I'm happy to answer questions, in real time or at the end. And now okay. So this is a slide that I use, a lot of people use, and it kind of, it's just here to make one point, kind of to frame the way we think about genetics. We think about genetic changes or genetic variants as having different frequencies and different effect sizes, right? So, what we call SNPs, single nucleotide polymorphisms, are common genetic variants with very, very low effect size. And that's what you can find in GWAS, genome-wide association studies. And then there are Mendelian disorders that we all know about, cystic fibrosis or maybe more to the point, dominant disorders that are very rare mutations, and the effect size is very high, right? And when you think about autism, because autism really is selected against, it's associated with reduced reproductive fitness or purifying selection, we're probably not going to find any autism genes that are both common. In fact, we know we're not, that are both common and high major effect, right? And of course, if things are very rare and have tiny effects, you're not going to find them either because you just don't have the power. So gene discovery in general in developmental disorders is kind of along this middle path where you're going to find common variants of weak effect, very rare mutations of major effect, and some things in between. So I'm going to talk about, in 2 slides, about common variants and spend the rest of the time talking about rare variants. And so for the common variants, and just to take a step back and put this in the context of autism, there's a lot of good work by multiple groups, and Bernie Devlin and Bert Clay wrote the first major paper on this that showed that most autism is caused by common variation or SNPs, and only a smaller fraction is caused by rare, highly penetrant mutations. But most of the discovery to date has been in this area here. Okay, so autism is a combination of common and rare variants, but most of what we find is in the rare variant space. So in the common variant space, this is the genome-wide association studies where you take thousands of individuals and you genotype them, and you look for differences in frequencies of these alleles. This is the work Jakob Grove published a couple of years ago, taking all the data that was available plus the data from ISAR, which is the Danish National Registry cohort, and doing a GWAS, genome-wide association study. So you have all the chromosomes here, and you have your p-value here, and things above this red line are significant. And so there are about 5 loci right now that have evidence for common variant association studies in autism. And when you have a locus like that, there are many genes under the peak, and so it's often very hard to find the exact gene. So this is still early days. What's happened over the past, since that publication, is between more data in the PGC, but more importantly, additional data from SPARC, which is the Simons Foundation initiative, and the Autism Sequencing Consortium that submitted 8,000 samples recently. There is a much bigger sample in the autism GWAS. And I would predict that sometime this year that data will be cleaned up and summarized and released, the results. But it's quite clear that there are many more findings in the bigger data set. So the data set's gotten about twice as big. It's still being cleaned in the PGC, but there will be more loci found this year. Okay, so that's all I'm going to say about common variation. So going now to rare variation, or things that are, again, rare but have a major effect. I'd like to mention the Autism Sequencing Consortium, which the names down there are some of the current leads of it. I dug this picture out recently because we've had a change in government in the United States, and some of us are happy about that. I'm one of those people. But the ASC, the Autism Sequencing Consortium, in a certain way started with these two gentlemen, because in 2009, they signed the America Recovery and Reinvestment Act, which was a stimulus package. And a small, tiny fraction of that money came to the NIH, and the NIH used it to kind of address things that were just not being addressed. And so, I don't know, this gentleman over here, Barack Obama, has since retired. And this gentleman over here came out of retirement to now be the current president. But in 2009, this was president and vice president, and they signed the ARA Act. And there was a specific call under the ARA Act from the NIH for groups that could, think about gene discovery and autism. And this group over here, Ben Neal and colleagues, got together. There were 5 sites that had samples and some sequence data, and it was a really strong collaboration between expert autism sites and expert sequencing sites. So 2 of the major sequencing centers, the Broad and Baylor, were involved very much so. And in the same time this was going on, the Simons Foundation generated a lot of sequence data, and 3 groups analyzed some of that data. So in 2012, 3 years after the signing of that act, these 4 papers came out with 1,000 families focusing on de novo mutations or new mutations, and 9 genes were found in that. If you put all the data together, you found 9 genes. So it was a really big deal. But if it takes 1,000 families to find 9 genes, it tells you that there are lots of genes to be found, right? And so, as these groups together began to think about the implications of these findings, we could begin to, Catherine Reuter particularly, and Bernie Devlin could begin to estimate how many genes are discoverable with this approach in autism, right? So, just like, when you see recurrent events, you can estimate how many separate genes there would be, right? If there's like 5 autism genes, we would have found them already. If there's 10,000, we probably wouldn't even found the 9 genes. And the best estimate remains that there are about 1,000 genes we can discover that have de novo or recent mutations of reasonable effect under a dominant model. And so, this was a white paper we published in 2012 that said with 1,000 genes, the 1,000 cases isn't going to get you very far. This is not even a significant finding. Greater than 2 de novo mutations is not enough in this sample size to be significant. You need 3 or 4 or 5. It depends a little bit on the gene size. So we said, look, we have 1,000 genes, we estimate, and we found 9 of them. If we really want to find more of those 1,000 genes, we need huge samples. And it's better to work together than to have a series of tiny papers and turn around and try and put them all together after the fact. And that paper in 2012 was the result of this meeting. This was the first meeting in 2010, where the Simons Foundation and the ARA group, so to speak, and other people as well, all came together and talked about this idea of sharing data, but pre-publication, trying to work together to kind of cut out that step. Usually, in genetics, we each publish our study on 100 families, then we put them together, and then we have 1,000 families, and that just seemed kind of counterproductive. So this meeting started this process, and then we met the next year at the NIH, and then we produced with a very large group, 150 people from all different perspectives. And then we wrote that white paper that said maybe we should try and work together as a kind of international entity. And, it's been, I think, pretty successful. We're now, it's been 10 years plus since that first meeting. We've had funding from the NIH for 8 years, and we're putting in for a new grant. And, I think of it as kind of building on 4 foundations: strong support, financial support, a lot of methods development, analytical methods development. So in fact, most of the money in the ASC actually goes to analyticals and bioinformatic resources. We have lots of collaborations. All the sites gave their DNA for sequencing. We collaborate with ISAR and other groups. And of course, there's ongoing improvements in the methodology of high throughput sequencing. And this really kind of brought us pretty far. This is the first ASC paper, now with a greater number of samples. And here we have 22 genes at an FDR of less than 0.05 and 107 genes with an FDR less than 0.3. And this is one of the early studies that showed that the genes in autism basically break down into things that involve synaptic function and things that involve the regulation of other genes. Okay. And then this is the paper that came out last year with now a much bigger sample set with 102 genes at an FDR of less than 0.1. And I'll talk a lot about this paper, but more about some of the things that aren't on the front page, so to speak. And again, just like before, this is also a Manhattan plot. And this is the evidence for a specific gene. But what's different between this Manhattan plot and the one I showed you before is when we say that SYNGAP1 is the gene we're studying is in fact SYNGAP1, because it's not a polymorphism somewhere near the gene that may or may not impact that gene. It's actually mutations in the coding region of that gene that we're counting towards the evidence for that gene. Okay. So here's the false discovery rate of less than 0.1. There are 102 genes above that line. There are, a good number that are genome-wide significant by traditional methodologies. And there are many, there's probably 20 of them that really, these are genes that have a few properties, right? The reason that a gene is all the way up here is because mutations are not rare. I mean, they're rare, but they're not ultra rare. And they're highly penetrant. You'll never see a mutation like that in a control. And those two things kind of will make genes gradually filter upwards. And the reality is that the relative risk of mutations in some of the genes up here can be as high as 20, 40, or even 80, depending on the type of mutation in the gene. So in some individuals, and many individuals with these mutations, it accounts for the majority of the risk. Doesn't mean there's not polygenic risk also, but if you took this mutation away, they probably would not have autism. So far, so good? All right. So, the ASC, that was last year's news. It's old news now. The Simons Foundation has invested in the so-called SPARK initiative, and they've released data now from 27,000 individuals. And all that data has been the ASC has accessed it. The ASC has actually added more samples of its own. And so right now, the ASC has a sample set that has twice as many cases as last year. So the paper from last year is half the size of the current data freeze. And now at a FDR of 0.05, so a more stringent FDR than I showed you before, there are about 105 genes, not 106. And this has been presented at ASHG and INSAR. Since it's not, I'd rather you heard the people who presented at talk, so I'll show you the link in a minute. But basically, it includes the Autism Sequencing Consortium, plus the data we had previously from the Simons Foundation, some new data from the Autism Sequencing Consortium, plus all this SPARK data. And that was one thing that changed since last year. And the other is that the tool that we use, the analytical tool that we use to identify genes called TADA, has been improved now 6 times. And the most recent improvements by Catherine Reuter and Bernie Devlin and others is, first of all, to include CNVs. So, you can take sequence data and actually call CNVs from it. So now TADA uses that data to bring in CNV calls. And so that's what so you see it says like, well, I can't, del and dupe. So now we bring in data from deletions and duplications. And it also has a more improved way of handling protein truncating variants and genes that are sensitive to that. So we can show that using the new analytical approach, we get more genes even from the last paper. But if I put those three papers, this is DeRubis et al., this is Satterstrom, and this is this new paper that'll probably, I think the first author will be Fu. So let's call it Fu et al. And these are different thresholds of significance, right? So this is a FDR 0.1. So right now at FDR 0.1, we have 185 genes in autism. Last year we had 70 something, and in 2014 we had a handful, right? So we're really it really is 1,000 genes. The interesting thing is that we're getting to a point where we probably could reasonably expect to capture in the next couple of years at least half of those genes because of all the sequencing that's going on. And the most shocking thing, perhaps for people who haven't followed this, is that we're not anywhere near a plateau, right? So it really is 1,000 genes, right? We're far away from like it leveling off and saying, okay, no more genes to be found this way, right? And again, I won't show you the list of genes. Not that everybody's happy to share that. But if you were able to go to INSAR this year, go to INSAR, register for INSAR. There were two presentations, one about the discovery side and the other about putting our data together with other developmental delay studies and see what's different or what's the same. And those links, they're active for 6 months after INSAR. So if you can see them there, they're short because INSAR talks are short. But it really goes through all the data and all the advances and then some of the interpretation, including showing that autism genes seem to, in aggregate, map to different layers of the cortex than other intellectual disability genes. So we're beginning to get some kind of functional differentiation. Okay, so what's the precision medicine or novel therapeutics? So I'm going to go back to the ASC paper and kind of pick apart some of the things that I think are worth considering. The first is, this is the 102 genes from last year from the ASC, right? And in the paper, we kind of loosely divide them into genes that are involved in neuronal communication, right? The green ones here, and genes that are involved in gene expression regulation. And then there's some cytoskeletal genes and some other genes as well. So the first thing, and this is already in the DeRubis paper and other papers by Van Eichler and others, there's clearly not just synaptic genes in autism. In fact, at least half the genes, plus or minus, seem to keep on being genes that are involved in regulating other genes. So, it's not going to be a simple thing, right? It's not just the synapse. It's, there's a lot going on there. I also mentioned, we've also spent some time and a lot in the most recent study trying to learn how genes that are more directly impacting the core features of autism differ from genes that have a broader impact on brain development. And so even if we take the ASC genes and split them, you can see that there's still plenty of genes that are autism predominant, in other words, mostly associated with social deficits and repetitive behaviors and not intellectual disability, for example, or motor abnormalities. And even most of those are these GER genes, right? So there was a thought that, first the thought was everything in autism is synaptic. That's not true. Then it was thought, okay, well, if there are some GER genes, maybe those genes have a more global impact. They're not autism specific. And, that's probably some truth to that, but, oh, I see a hand. How do I?
Adolfo Garcia. Thanks. It's just asking you, I can see here P10. This is a very broad action phosphatase of phosphatidyl inositol triphosphate.
Yeah.
It has a lot of implications in cancer. Why do you put it in the ESD predominant? P10.
So P10, P10, there is a syndrome, as you know, P10 Hartoma syndrome, that that actually a non-trivial fraction of those individuals meet criteria for autism. The ASC, one of the strengths of the ASC or the weaknesses is that it's a very, very broad net. And so samples came from many places, some places that were strict academic sites that only, collected autism, like, more idiopathic autism. Some were clinical sites, some were, convenience samples and so on. And so, some sites probably would have, if they would have seen somebody with, with P10 syndrome, they may have excluded them, but some places they probably didn't even know that they had the syndrome. There are a lot of genes on this list that have a cancer association, in fact. And I think that's probably because a lot of these genes are developmental and they're not purely kind of synaptic, right? They're involved in regulating other genes. And I mean, FOXP1 is, mutated in cancer, DDX3X is mutated in cancer, so the cancer autism link, I think, is real. I don't think it's a main driver, but many of these genes have this dual role that they could be found in cancer and in and lead to abnormal development. And sometimes it's different mutations. Some of the genes, like DDX3X, if it's constitutively active, you can get a cancer phenotype. So it's a somatic mutation there. But if it's inactivated, then in kind of in a germline way, you might get autism and developmental delay. I don't know. I hope that answers the question. I couldn't hear all of it, but
you answered the question. Thank you very much. May I ask you another silly question?
No such thing. Yeah, go ahead.
The autism spectrum is enormous, and from a clinical point of view, it is rather complex to define who is within this spectrum. Are you selecting those patients that have only very dramatic forms of disease, or you are taking the whole population? I'm not very familiar with the classification because when I studied medicine, autism was non-existent essentially. Then everybody was diagnosed with some kind of autism. Because if you were shy, you were Asperger type and things like that. What are the clinical criteria to be included in your question? And I promise I won't ask anything more.
Oh, this is a great question. These are great questions. And again, it's back to the fact that we have a lot of different contributing sites. I would say that at the beginning, everybody had to have an ADI and ADOS. That was kind of like, because it was driven by the state of diagnosis at the time. So there was a ADI, a questionnaire to the parents, and an ADOS, an observation of the child or the individual with autism. Some sites, they were expert sites that would spend, maybe they wouldn't do the full ADI, but they would spend 3 days with the family, and they were expert clinicians in autism, and so we took their diagnosis as well. Some sites, like we have a study going on in Sweden that uses a national registry and uses clinical diagnoses, right? What we found, and I think you're kind of getting at this, is that, when you look at more severely affected individuals, you're more likely to find a mutation. But we have a lot of data from less severely affected individuals as well, because it's such a conglomerate of collection sites. And we still see the presence of mutations, even in individuals that are at the high end of the spectrum. I wouldn't call them, I don't think anybody ascertained only, let's call it Asperger's, like high functioning Asperger's. Some of the samples clearly include that, but most sites cover the spectrum. And I would say there is a bias in our cohort towards individuals that are more severely affected. So they definitely have ADI and ADOS criteria, and they may have other things as well, comorbidities or intellectual disability. So if we are sampling, a subpart of the spectrum, it will tend to be the more severe side. And maybe I have a figure that, well, I actually took that part out. So maybe I'll just, an easy way to put all this together would be if these genes in purple regulated these genes in green, and you can go back and say it's all about the synapse, right? And there's a way, since many of these GER genes, we know they're targets, we could actually ask if the targets of these genes are enriched for these genes. And this is one of those very difficult to understand figures in the paper, but basically it says, here's a bunch of GER genes that regulate other genes where the targets are known, and they show no evidence of regulating the synaptic genes. They show evidence of regulating other genes that are GER genes where the targets are not known. So there's a very high, these do a really good job of regulating these, that's the p-value there, but there's really not any good evidence that they regulate the synaptic genes. And so it seems like a trivial point, but it actually really was an important one. It said that the GERs are not solely working through regulating synaptic genes. So again, it's not a simple pathway here. Another thing we did, there's more and more work in single cell data. And so this is just kind of looking at inhibitory neurons over the course of their development. And this is excitatory neurons plotted out over the course of their development from very early to fully mature, early to mature. And using these specific plots. And the autism genes, what you can see is they map very nicely to late inhibitory neurons, but they were up really nicely to late excitatory neurons, right? And even intermediate excitatory, intermediate. So, it's also not just excitatory or just inhibitory. And this is just a statement that we've, more and more in genetics, we understand that many genes follow a U-shaped curve. In other words, some people call it Goldilocks. Too little is not good and too much is not good. You need to be just in the middle. We already knew that from copy number variants. Like this is the de Jour syndrome locus, 22q11, probably the best known, historically best known copy number variant. And both the deletion and the duplication, which arise kind of just by the fact of the way that the deletion happens, you can get a duplication as well. Both have phenotypes, so too much or too little. Same true for the Williams syndrome locus, for Smith-Magenis, for 16p, even Shank 3 that some of you have worked on, right? There are a non-trivial number of families where there's a balanced translocation in a parent that can lead to monosomy, disomy, or trisomy in the child. And there is a phenotype in the monosomy, this is Phelan-McDermott syndrome, but there's also a phenotype in the trisomy individual. So 3 copies of Shank 3 will cause a phenotype, right? And that's been pretty well documented. So, you can't just add more Shank 3. You have to kind of tighter what you put back in if you're thinking along those lines. Okay, and this is back to the question we just dealt with a little bit. If you look at all the mutations, all these genes and their mutations and the associated phenotypes, the tendency is that the carriers of these mutations tend to be more severe. So this is, again, Phelan-McDermott syndrome. These are deletions. This is a first study which we put out a few years back, looking at individuals with Phelan-McDermott syndrome with deletions. And you look at the IQ distribution. I mean, 50% had profound ID. Okay. Now, it's a biased sample. Genetics is most commonly done on severely affected individuals. We can go on, but there's a real need here. And this is Shank 3 point mutations. And again, we brought in a lot of individuals with point mutations. And, they do have intellectual disability and they have language delay. It's not just autism, right? So it's a pretty profound phenotype. On the plus side of the ledger, if you will, is that the genes we're finding, it's partly because of the design we're using, are mostly loss of function dominant genes, right? In other words, they're on the autosome, there are 2 copies and 1 is destroyed. What that means is there's another copy that's still functional, right? And so, if there was a way kind of generically, and there are ways, right, to think about turning on not one gene at a time, but developing general methods like CRISPR, CRISPR-A as an example. All the genes that are being found in autism, intellectual disability, more than 80% of them are loss of function dominant mutations. And so, you could think about like more of a platform approach to how to, address a missing gene if you knew how to turn a gene back on. There are, of course, genes where almost everything we see is a missense. Like these genes over here have no loss of function variants, and everything is a missense variant. And these genes are almost certainly gain of function mutations. So the missense mutation is not killing the protein; it's making it do something different, right? So all this is plotting how many loss of function mutations do we find versus how many missense mutations do we find. And you can see for the most part, lots of loss of function, but there are a few genes where it's a zero here, and and sometimes it's probably by chance if there's just two events. But when you get up to DEF1 SLC6A1, there are just too many. It is actually a gain of function. It's actually been shown by other groups. KCNQ3 is a gain of function. SNC1A is a gain of function. All right, and this is another on the plus column. There are lots of papers that show if you take some of these genes and turn them back on, you get recovery. It may not be perfect recovery, but it's some recovery. And Guoping, did a really elegant study where his group turned Shank3 back on at different times in development, including in adulthood, and compared the recovery you got when you turn it back on in a young mouse versus an old mouse. And that really was an important study because most people weren't so careful. And the point was that when you turned it on, even in the adult mouse, you got recovery. It wasn't as good as earlier, but there was recovery nonetheless. I'll just remind people that there's been some comments on the methodology that we've all been using that Speeded Out pointed out that some of the completely neutral constructs we use may have a phenotype, but there's also a follow-up by that same group that shows that even when you correct for that, there's a finding there. And it's not just Shank3, it's been shown for actually FMR1, it's been shown for many genes where, at least in a mouse, you turn them back on a little bit late in development, you still get recovery. Okay, and here again on the plus column, if you will, is that these genes that we're talking about are relatively common. What that means is they're rare, but they're not ultra rare. In other words, if you said, I'm interested in studying Shank3 patients, they're out there, right? And some of the genes, are in the order of 0.5% or 1% of all autism, 0.5 or 1% of all intellectual disability. And that's not a bad place to be. You can actually find those individuals, right? And pharma loves this because it also gives them under what they call orphan designation. They get extra protections if they go forward with this. Okay. And the other thing, as I mentioned before, is that the top genes that we're finding are highly penetrant. They account for most of the risk, right? They have odds ratios of 20 or 40 or more. Okay. So I just showed you a bunch of slides, and this is what I was trying to say. That when you think about gene mutations in autism, intellectual disability, and developmental delay, these are things that we think we know to be true. There's not a single pathway. There's just no way you could convince me right now there's a single pathway to autism. So suddenly you kind of have to think about precision medicine or targeted therapeutics, or you name it, right? You have to think about, how do I tailor the treatment to the biology that's in front of me, not to autism, but to the biology that's causing that autism. They're overwhelmingly dominant, but again, that's partly an artifact of how we're finding them right now. But it does say that, for all these genes we've talked about, there's a second allele for many of them that can be manipulated as a target. They're primarily loss of function, and we tend to know the mechanism. In other words, we've seen enough mutations in these genes that we can say this is a gain of function, this is a loss of function, this is a mutation in a key domain, and other mutations outside that domain won't have an impact. So, we're learning about the biology just by having a lot of events, a lot of mutations. It's unfortunate, but it's true. It's almost certainly the case that many of these genes will show a U-shaped curve. You can't just turn them back on and hope for the best. The genes we're finding, the top genes, it's severe autism. It's autism with profound autism, intellectual disability, language delay, and motor delay. There's a real need there. This is not, we don't need, this is not a highly functioning individual that, probably doesn't need our help. Obviously may need some social help and psychological help. We all need that. These are people with very, very profound needs. And so you really do think about, are there therapeutics that we can bring to bear sooner or later? And as I showed you, for many of the genes that have come up, it has been shown that turning it back on, after birth can be helpful, because this is one of the questions we often get, which is, well, is this locked? Since, these mutations happen at conception, is it irreversible? And first of all, we know it's not irreversible completely because you can do behavioral interventions, but also in the preclinical space, you could turn the genes back on and you see recovery. And then, as I said, the top genes are rare, but not ultra rare, and they're highly penetrant, which means, you can, you kind of know that mutation and whatever it's doing downstream is accounting for most of the phenotype. So it makes it easy to think about, a mouse model or a stem cell model, because there's a direct relationship between the mutation and the phenotype. All right. So really, when I talk about precision medicine autism, it is my perspective that there's really not any alternative, that we're not going to, I kind of said that already, but this was, this raised some eyebrows when we was first presented a few years ago. There's not going to be a magic medicine for autism, right? All right, so how do you get from here to there? So in the next few minutes, I'll talk about the way we perceive what our contribution can be as an academic medical center, and also what's missing, right? So, the first question is, what's your target? And, if we have the ASC data or other, any data anywhere, we can really say, okay, I'm interested in this gene or that gene or the other gene. And then obviously, nowadays, you can make neurons, human neurons, particularly from patients. So you can, if you're interested in Shank 3 and you have families with Phelan-McDermott syndrome and you draw blood from them, or you can make human neurons in culture and you can begin to study the neuron carrying the mutation. Of course, we can make rodent models of that same gene, and we can see patients with that same gene, right? So we can make this kind of nice circle where, from the point of view of drug development, you can identify your target and you can perhaps create high throughput screens. You can mutate the gene in vitro, look for a phenotype, and then try drugs. Or you can, you can develop conceptually, say, well, this is an enzyme. What happens if I stimulate the enzyme, right? And so, and you can take your neurons in culture and you can make reasonable assays, both for high throughput, but also for high content. So you can say that this compound is working or not working. You can, of course, once you see a phenotype in your mice, you can try that compound in the mouse. And then, of course, you can go back to your patient population to clinical trials. So this is, I think, what we as academics can do very well. And we, people who are more in biotech and pharma, what they do very well is these screens over here, once there's a target, coming up with a screen-to-screen libraries and to validate compounds. And then if something makes it all the way over here, to do the medicinal chemistry to actually make it useful, appropriate for a patient trial. So, we're trying to push this part with the hope that we'll find partners sooner or later in kind of the more biotech industry space that would be interested in developing high throughput screens, screening libraries, and then doing medicinal chemistry. So like to give you, the main genes we're working on are ADMP, DDX3X, FOXP1, and Shank3, and we've added CHAMP1 and we're adding Shank2 as other genes. And because, we're very lazy people, we go for genes that, are top of the list. We're not trying to make our lives too difficult. DDX3X is not on this because it's on the X chromosome. It's a very interesting gene I'll talk about very briefly at the end. But FOXP1, ADMP, Shank3, DDX3X, those are the genes we spent a lot of time on. Shank3, Violetta can tell you all about it, and Maria as well. It's actually been known that there's something going on the terminal end of chromosome 22. It's been known for over 50 years, right? So when chromosome 22 forms a ring, which it can do, you lose a little bit of genetic material at the very ends. So ring 22 was easy to identify even with low resolution karyotyping. So that's why we know about it 50 years ago. And the phenotypes associated with ring 22 are intellectual disability, motor abnormalities, language delay, all the things that we associate now with Phelan-McDermott syndrome. This is the paper I just showed. This is the paper that showed Shank 3 mutations in autism. So this was already, this has already been seen in the literature. This has already been seen. This is the deletion. It's this point mutation in these brothers that really kind of showed that, mutations in Shank 3 were enough to lead to an autism phenotype. So an important paper. And then, we've been kind of systematically, we've seen over 100 patients now at the Seabra Center and across a larger network too. And from about 10 of those patients, we developed IPSCs. And so we're beginning to map, these are human neurons in culture, beginning to map some of the dysregulation of pathways. I won't get into it. We and others have made now 13 different mouse models of Shank 3. And in some ways, these are different models here. Many of them show motor deficits. I can't even see my own writing. Many of the mouse models show social deficits. Many of them show excess repetitive behaviors. So there is some consistency in findings. And when you have these mice and you have, you see something, behaviorally, you might be able to, this is electrophysiological. This is LTP, long-term potentiation. In a wild type mouse, you get induction of LTP and maintenance. If you're missing one copy of Shank 3, you get the induction, but the maintenance goes away very quickly. If you give IGF-1, and we could talk about why we chose that, you preserve the LTP, right? In the rats we made, if we gave them oxytocin, we preserved, they showed deficits in both attention and social recognition memory, and both of those were reversed with oxytocin treatment. And as I said, we've also seen lots of patients, right? And so now we can take all that together, the in vitro work, the animal work, and the patient work. This is more patient work. And we can start to try those same drugs. IGF-1 was tried in the mouse and shows preliminary efficacy. It wasn't really an efficacy trial, but safety trial in PMS. And we do similar things with other drugs. So we're trying to get from now we're using drugs that are already very safe and known to be and safe in kids, so we don't have to go through the entire drug discovery pipeline. But the idea is to even take some existing compounds and see if they might work in the preclinical models and then the clinical models. And the last example, which we're just now publishing a bunch of, is DDX3X. I mentioned this is an X chromosome gene. What's remarkable about it is, first of all, it's almost only found in women. Men or boys with the mutation probably don't survive. This is MECP2. MECP2 is the cause of Rett syndrome, which many of you might have heard about. It turns out that DDX3X is actually more common than Rett syndrome, and probably a lot more common than Rett syndrome. This is data from the DDD of a serially ascertained cohort of 12,000 individuals. And this is the excess of mutations in MECP2 in this cohort, and this is the excess of mutations of DDX3X in this cohort. So it's at least as common as Rett syndrome, and it's probably, nobody even heard of it 3 years ago or 5 years ago. And like I said, it primarily affects girls because boys can't carry a loss of function mutation in this gene. And just like I showed you with Phelan-McDermott syndrome, we've only seen 15 in person because of COVID, but this paper just came out. This is kind of back, we do a lot, we look at their intellectual function, adaptive function, motor functioning, language, ASD, sensory, behavioral, and so on. And so we're beginning to map out the phenotype with DDX3X syndrome. And here too, we take samples, blood samples, and we make IPSCs. One of the interesting things about this syndrome is that if you look at, we have both in blue missense mutations and in brown nonsense mutations. The phenotype tends to be more severe with the missense mutations than the nonsense. So this is a gene that actually has 3 different phenotypes, 3 different severities of phenotype based on the kind of mutation. And actually the loss of function is not the worst one. The worst one is actually some gain of function mutations that are buried in the missense. But that's online. This just came out. And then, and the senior author is Dorothy Grice. And then Sylvia de Roubaix just got a paper accepted where she and her team knocked out this gene in mice and did some phenotyping. And so it's in bioRxiv on the bottom right, but it's also, it'll be out probably in a few months. But bioRxiv is so good. I can't. But what you can see, for example, is when you cross these mice together, you see no males with the mutation. They don't survive, right? So the males will be right here. And again, it's all online now, but there's some evidence for ectopic kind of cortical laminar abnormalities in these mice. And there are behavioral deficits in milestones. They show hyperactivity and anxiety, deficits in memory and motor impairments, and probably some later life decline. So, it's kind of the same thing. We think, we're using data from the ASC and other groups to figure out molecular targets. We're trying to work up human neurons and animal models and also see patients. And then the idea being to kind of, help facilitate this lower thing, which is this drug discovery pipeline. Okay. And we can talk about, can we really do that for 1,000 genes? And that's why we go back to this idea of platforms. Can we develop a strategy that work for many genes? But maybe that's more for the discussion. So maybe I probably should wrap up anyways, but I want to thank you all for listening. And I hope I kind of gave you our perspective as a center, but also as a, also the genetics that represents a lot, the International Consortium. And I'll stop there. I'll stop sharing, and I'm happy to take questions.
Thank you very much. It was a great talk. I want to ask the audience in Zoom if you have any questions before taking some in YouTube. Okay, I will take, okay, go ahead, Ivan.
Hello, thank you for your talk. I know that this is not work from you, but I would like to get your perspective. You mentioned that the group of Dr. Guoping Feng showed that you can restore function of Shank 3 and some of the symptoms go away. How do you reconcile that with the idea of autism being a neurodevelopmental disorder?
Well, I mean, a lot of things are neurodevelopmental, but it doesn't mean they're, written in stone, right? I mean, if you just look at what does work for autism, like behavioral interventions, right? When people study like, ABA therapies and autism and say that it helps half the kids, they don't really look at the cause, right? They don't go back and say, this was a Shank 3 mutation, right? So behavioral interventions are an example of things that work, in some ways independent of the exact mutation, right? So, there is some plasticity in the human brain, and that's what I think ABA or behavioral interventions are capturing. I think, but I do think even, it's almost certainly the case that for most of these mutations that not everything happened in the first, week of conception, right? When you look at, the Rett syndrome gene, the Fragile X gene, at least in mice, you turn it back on, or in other animals too, things get better. They probably don't get better. It probably would have been best never to lose it in the first place. But if you turn it back on, it gets better. And I mean, what Guoping showed is that turning on earlier is better than turning it on later, because we lose plasticity over time. But we are, the young brain is quite plastic. Right? So I think that's what's happening. It's a neurodevelopmental disorder, but they have plasticity that can correct something. Can it correct everything? I'm sure that if if the cerebral cortex is not properly gyrated or neurons are absolutely the wrong place, you're probably not going to see a reversal. But for many of these things, it's probably less severe structural changes.
Okay, thank you.
Well, I will read some of the questions in YouTube. Veronica Ati Morales asked if we are talking about a neurodevelopmental disorder, I'm guessing therapy would have to be a very early intervention, right? Otherwise, it would be hard to recover development.
Yeah,
I guess, early intervention.
Yeah, so I think, I think the reason I showed Ping's paper and there are many other papers like that is because this is one of the most frequent questions you get when you think about therapeutics, right? When is the right time? And I think the answer is we don't know, but for some of these genes, it's probably going to be that you can intervene later and still see a very positive effect. Some of them you're going to have to intervene very early to see the best effect. And that makes it very hard because if you have to intervene at day one, nobody's going to do that, right? But I think the way I think about these things is we're not trying to cure them. We're trying to make things better, right? And if somebody who's 13 years old and has intellectual disability, motor abnormalities, language delay, and autism, severe anxiety, and so on and so forth, maybe if we treat them when they're 13, we can reduce some of that. And maybe if we reduce them at 3, we could reduce more of that. But I'll take the 13-year-old, right? And certainly, if it works in a 13-year-old, then it becomes very easy to go forward and try younger and younger ages, just like we did with the vaccines, right? It was first in adults, and older adults. Now suddenly, some countries are doing 12 and up, and so on. So, if we can show efficacy, some positive, a small positive change in the, adolescent group, if you will, then of course people will want to try younger and younger because it's probably going to do better. It's probably have a better impact on younger and younger. But is it going to have no impact on adolescents? I'm sure that some of these genes, not all of them, some of these genes will be able to reverse some of the deficits, even in adolescence. And again, I go to the ABA story of behavioral interventions. We know that if you do behavioral intervention, even in a 30-year-old, you can get some benefits. Of course, it's much better in the first 3 years of life or the first 5 years of life, right? So, we have plasticity. We can make changes, but no question, early is better.
Exactly. Some help is better than no help at all.
Yes.
Other question says that, did you find mutations in genes involved in lipid metabolism?
Well, almost. It depends on exactly how you define it, but I mean, some of the genes we found are directly or indirectly involved in lipid metabolism. I'm sure there are some that are direct. I'm just not an expert, but the gene lists are, they're available, certainly in the published paper, you can take a look.
Thank you. And another question from Aliesha, the same person. How is BDNF expression in autism?
Yeah, so that's a great question. I mean, one of the complexities of autism, well, there's 2 or 3 right now. One is it's highly heterogeneous, right? So BDNF, it's an hypothesis, may be up in this individual, but way down in that individual and unchanged in the third individual, not because BDNF is not important, but because autism is very variable biologically. So that's one problem. And the other problem, of course, is that there is a small universe of postmortem samples, many of which historically were quite old and poorly preserved. And most studies with autism, like looking at postmortem samples, they have an N of 20 or 30 or 40, which is a small N, relatively speaking, especially when you realize that autism is not one thing, right? And so you may, again, if you have 20 people, I mean, look at the neuroimaging field, and historically people did neuroimaging in 20 individuals with autism, and they saw something very clear, but they didn't see it in the next 20 because the next 20 were biologically different, right? And so the postmortem studies, because there's just so few samples, they've never really it's hard to make a consistent message. So what people are doing instead is, if they're interested in a gene that's mutated in autism, they might look at the mice and see if, in a specific biological subtype of autism, is BDNF changing. That's a different question. And there are some evidence in both directions.
Okay, thank you. Yasmin, that is also another researcher that works with one of your models, actually, Shank from your lab. She wants to ask a question. Go ahead, Yasmin.
Thank you. Thank you, Giselle. Thank you, Dr. Buxbaum, for the talk. I want to ask you something. The insulin-like growth factor, oxytocin, in the treatment for kids, how physiologically that can be work? What they are doing? Because in a paper in IPSC cells, they, with the insulin-like growth factor, they showed that probably they can induce like a mature synapses. But to me, is something that I don't know. Is it real? Maybe because in your model, for example, the synapses structurally doesn't look like immature. So, what do you think that this treatment is doing?
Yeah. So the short answer is these treatments weren't chosen because they're great biological tools to reverse Shank3. These treatments were chosen because the families are desperate and they're very supportive of drug repurposing. If there's a very, very safe compound out there that may have some slight positive impact, we would want to try it, right? So the IGF-1, we chose IGF-1 for A, very safe, B, it's used in kids, and its known effects, if you will, and they're poorly mechanistically described, are like when you inject IGF-1 into rodents, it does seem to push synaptic, like the PSD, PSDs get bigger. I mean, it's really weak. This is old data, right? But it does seem to push glutamatergic synapses towards being more stable and mature. So that was the rationale. Is it a great rationale? No, of course not. And the mechanism is really opaque, quite honestly.
Okay.
But it was something that there was evidence that there were immature neurons, at least in our hands and other people's hands. And IGF-1 is known to promote maturation of neurons through unspecified mechanisms. And it's also very safe and used in kids. So that was the rationale. It was a very, the neuroscience behind it was the weakest part. Let's put it that way.
Okay.
And with oxytocin, it's a little bit better because social recognition memory, actually involves oxytocin directly and other neuropeptides. And so that wasn't such a stretch, right, to try that. We didn't actually choose oxytocin in that model because oxytocin is the love drug. We chose it because actually social recognition memory involves oxytocinergic neurons. But again, if you give oxytocin peripherally to an individual, for the rats, we actually inject it into the brain. So it's a different modus of.
Yeah. I see. Thank you.
Dr. Adolfo, if you want to ask.
Yes, I'm just curious. Geneticists usually don't study only the patient, but also the relatives. I'm sure you have plenty of data. And have you been able to study twins with the syndrome or siblings, very related to the patient, and some specific genes to give a clear picture?
Yeah.
What's going on?
Yes, I mean, we don't focus as much as the siblings as we should, but it also depends on the study, right? So when we and others are looking at inherited variants, common variants or rare inherited variants, then people do look a lot at the siblings for phenotypes. Most of the genes that we're talking about today, the mutation is de novo. So the sibling doesn't carry it, right? One of the things that one of the questions that's being asked now is, if autism is a mixture of some people with primarily, rare mutations, some people with primarily polygenic risk, and some people in between, what does that look like if you look at the polygenic risk in individuals with or without mutations? And actually, it's quite interesting, because if individuals without the mutation, autism without the mutation has this much polygenic risk. Autism with a mutation may have less polygenic risk, right? Because the risk is also coming from a combination of the polygenic risk and the rare mutation. But when you look at polygenic risk in siblings, it actually is, all the siblings tend to share, I guess, predictably, a lot of polygenic risk, because, if the family has 100 loci in mom and dad that are contributing risk, then all the kids get some of it, right? But with the rare de novo mutations, mom and dad don't have it, and the siblings don't have it either. But then the new space that people are looking at are rare inherited mutations, which have a bigger effect size than the de novo, than the polygenic, the SNPs, but less than the de novo mutations. And so, it's kind of the next thing people are targeting, the inherited rare variants. And then it'll be very interesting to look at what you see in siblings that carry that, but may not meet criteria for autism.
Thank you very much.
There are a couple more questions from our audience in YouTube. Carlos Chicalote asks, do you think that regulation in the glutamatergic synapse can change the phenotype?
Yeah, I think it can, but I also think we have to be careful to make sure that the person you're treating has that deficit, or has that, I think that's the most, the most significant thing to come out of this past decade, is that autism is really a bunch of different things. And, it may be possible that, regulating glutamatergic synapses will completely cure 5% of individuals. But if you did if you didn't pre-select those individuals in advance, you may not see the signal, right? Because it may not work at all in 95%. Or it may be bad for 95%. Remember that even with the same gene, too much or too little can be bad, right? So, precision medicine is that's the issue, that the drug may only work in some people, may be harmful for other people, and it may have no effect in the third group. And so there's probably no mechanism that you all collectively can think of that wouldn't probably be a great idea to pursue. But you have to kind of embrace that it may only work for a subset. So how do you find that subset? It may be deleterious for a subset. So how do you avoid that? And then it may have no effect on a subset. And so, don't do your clinical trial in a mixed population where you're going to, make some people sicker, some people better, and some people no change. You may not, it wouldn't be very good.
Of course. And another question is, a recent study found a higher rate of somatic mutations that appear in early development of autistic individuals. What do we know about the contribution of these kind of variations to ASD?
Yeah, I'm not that familiar with the somatic mutation literature. Obviously, again, part of it, of course, is, if you're interested in somatic mutations, you really want to study brains rather than periphery. So that's always a limitation of studies in somatic, and again, there aren't too many brains available. But what you can see in the periphery is that quite clearly there are some mutations that are being maintained, so to speak, as with some basically mosaicism, right? And the reason I say maintained is because it's probably the case that some mutations are so deleterious that they're not really compatible with development, or they're so severely affected. And the reason those individuals come into an autism study is because they are mosaic, right? So they have post-zygotic mosaicism or somatic mutation, and that protects them from the worst effects because they're mosaic of that mutation. And so you discover it because they didn't die, basically. So I don't know if, I'm not really convinced that there's excess, that there's clearly excess mosaicism in that sense, but I don't think it's a major driver of autism. The mosaic is protecting those people from a more serious outcome.
Mm-hmm. Yeah, that makes sense. Well, one more says, well, there is one that says, do you know if there is a list of top genes with common variants relevant to ASD?
Well, as I said, there's only, the Grove paper has sort of like 5 loci, right? And that's the top loci. But the problem is, and in the Grove paper, it'll mention the closest gene, but we know from GWAS studies that hundreds of genes are under each peak, and you don't really know what is the gene that's contributing to that peak. So the short answer is, there's a list of genes, a very short list, but it's actually not, it's not the driver gene, it's just the closest gene to the peak.
And one last question from YouTube. What is the potential for rare genes associated with autism to use in gene therapy?
Yeah, I think that's where, I think the answer to that is in the future, that'll almost certainly be the approach, right? I mean, I think with the huge breakthroughs the past 2 years in CNS disorders and gene therapy, particularly spinal muscular atrophy, that's the wave of the future. And the thing about gene therapy is, it's not just virus expressing protein, right? It could be an antisense oligonucleotide, right? So if, one of the genes that come up a lot are the sodium channels. Right? And some of the sodium channels, with an antisense oligonucleotide, you can, because it's a gain of function, you can actually, in preclinical models, you can like make things better by using an antisense to block the gain of function mutation. And that's already in clinical trials, right? So gene therapy doesn't mean just AAV plus cDNA. It can be antisense oligonucleotides. It could be, targeting the 3' UTR. And I think that those are I think we're going to see some examples of those in the autism space in the next few years, less than 5. I mean, we already are.
Yeah.
We're trying it in Rett syndrome. And UBE3A is already being tried for gene therapy approaches.
Yeah, it is coming up on us. It's very fast, I think. And this is all the questions. I just want to know your perspective. I know there are many pathways that could be affected, and that's what the data shows very clearly. But do you think there is a convergence in the mechanisms that are affected in the brain, actually? Because at the end, the symptoms are shared among different patients. I know it's a spectrum, but nevertheless, we see that there are symptoms that are very present, and probably the circuits that are underlying these symptoms are very similar. So at the end of the day, we have certain, come back to certain structures, right? So what do you think about that?
I think it's probably good to keep an open mind. I'm not convinced that there's going to be clear convergence to a single pathway. I mean, what people are trying right now is they're trying to like knock down all these genes like a high throughput methodology and look at common downstream, pathway changes. But nobody believes there's going to be one. People are saying maybe with 100 I could find 10, right? I mean, if your car doesn't run, I mean, is autism like a minor change, or is it a significant, like a core change? Remember, if autism involves at least 1,000 genes, and we only have 20,000 of them, it's a pretty basic, it's a fairly broad and basic thing that's being disrupted, right? So, if your car doesn't run, is it because the wheels fell off, or is it because the engine fell out, or because there's no gas in the tank? I mean, in the end, of course, it's all the drivetrain, but I don't think we're gonna see easily common pathways. We already know that, that striatum is implicated in FOXP1 and in Shank 3, but it's clearly not implicated with many other genes.
Of course. Yeah, the same. I was thinking same as if we can help some of the symptoms, maybe it's better than not have any treatments, right? I don't know if the audience have another question. It seems that we don't have any more questions, but we will be very happy if in the future you can visit. If it was different, we could take you to eat some really nice Mexican food, but unfortunately with the virtual world it's impossible. But it was such a nice talk, and thank you very much for showing us what the Cyber Center is doing. I think it is a very nice model to follow, and it should be implemented, because I guess the populations that you are studying are very particular, and different populations should have also a probably different mutations, right? Like you're probably more focused on the Caucasic population, probably.
The de novo mutations, because they're de novo, they tend to be pretty universal. But we are very happy to work with other groups around sequencing samples from diverse populations. In fact, we have a new grant that we submitted to increase representation of Central and South American countries.
Oh, that's great. I hope it is approved. Yeah, thank you very much, Joseph, and thank you everybody for listening. And we will be in contact with you. We'll be very happy to talk and share also our data.
Yeah. Well, thank you again for the invitation, and thank you for staying on so long, everybody.
Thank you. Bye. Take care.
Have a great weekend.
Same to you.
Thank you very much. Bye.